Hello viewers, welcome yet again to our ongoing series in genetics entitled Mendel and Beyond. Now in the past few lectures, we have devoted ourselves to discuss many of the deviations which arise from Mendelism. In the last lecture, we had a very detailed discussion on the concepts of linkage, how it was discovered, and in today's lecture we are talking about a mere extension of linkage. As you would appreciate, learners, linkage and crossing over can never be divorced. They would always stay together. So we always use the term that even linkage and crossing over are linked forever. Let us talk about how the whole process of crossing over was discovered. But then, as I said, without linkage, crossing over could not have been discovered at all. Just to recapitulate a few fundamental points, the Mendel's experiments had earlier shown that actually the allelic pairs for genes which were influencing, say, different P characteristics, for example, size and the color, they had a characteristic to get assorted independently. And once we saw that this independent assortment existed, naturally it gave rise to very precise ratios of the progeny. We had also concluded so many times that 9:3:3:1 would be the phenotypic ratio, and of course the other genotypic ratio, which was a very long one, 1:2:4, 1:1:1, was a dictum for dihybrid cross. So this would mean that when we are talking about independent assortment, there is naturally a pairing between the different genes. Why call it genes? First, let us begin with the chromosomes, and then after this pairing, the segregation would come between the chromosomes. And when it comes to independent assortment, then naturally A could go with capital B, A could go with small b, and likewise small a could now get combined with capital B and small a and capital A with small b. So that means all various kinds of permutations could be achieved in this case. So in other words, the genes which are influencing the color and the texture, they definitely are residing on different chromosomes, and therefore the different chromosomes or the chromosomal pairs, they are going to behave independently of each other at meiosis. What do we wish to say here? We wish to signify that the color, its inheritance is going to be independent of that of texture and vice versa. But then we are going to ask ourselves a question: do such allelic pairs show regularities in their patterns of inheritance always? And is there any diagnostic ratio or other pattern also which is associated with such genes? Or is it that the independent assortment is universal and cannot be changed at all? Yes, the answer to the question is an affirmative, and that is, there are in fact simple patterns which are unique for genes which are residing on the same chromosome. Now viewers, please understand, we are talking about the genes which are located on the same chromosome showing even different patterns. Let us take a small example. There is a dihybrid, which is an individual which happens to be heterozygous for two genes, say capital A and small a, of course, the allelic form, and capital B and small b. Now these genes, let us suppose they are located on the same chromosome. How do we represent this chromosomal complement? As you can see on your screens, capital A and capital B on one chromosome and small a and small b on another. So what are the results that we would get in the inheritance? We would find that rather than sorting independently, these capital A and capital B would in all probability would be inherited together. and likewise small a and small b will also be inherited together. So, in other words, how can we assume that now the inheritance pattern in the result would be the same as that of 9331? Definitely, this inheritance pattern will be different from those resulting from independent assortment. So, in such cases, we would find that maybe the fragments of the chromosome gets broken, and if it is so, then the parental chromosomes will have a situation where capital A will get inherited along with small b, and the small a will get inherited along with capital B. So, there seems to be some sort of a mechanism where there is a precise breakage and union of the parental chromatids during the process of meiosis. So, learners, it is precisely this cut and paste process of the fragments of the chromatids which is called as crossing over. And when does crossing over take place? It takes place when there are 4 chromatids which are produced as a result of the duplication of a chromosome pair, and it is quite customary to call this group of 4 as a tetrad. And it is these crossover events which are not predetermined. They are going to occur more or less randomly along the tetrad, and in some myocytes they will be in certain positions, in other myocytes they may assume this process at different positions. So, that means if one was to look at a snapshot of different myocytes which will show how crossing over will take place at different places and in different ways, and this is how it would distribute itself in the among the 4 chromatids. So many combinations are possible. In other words, new combinations of alleles will arise from these crossovers. On your screens you would find a situation where we have the 4 chromatids. Of course, 2 of them are the sister ones and the 2 other ones are also sister, and among these they would be, that is in relation to each other, they would be non-sister. As you can see, the exchange of segments is between the non-sister chromatids. So, it could be between the 2nd and the 4th, it could be between the 2nd and the 3rd, it could have a crossing over say between 2nd and 3rd at one place and the 2nd and 4th at another. This would be some type of a double crossover, and it could also be a multiple crossover. So, definitely how can you assume and how can you expect that the 9331 ratio would be got at this case? So, that means crossing over is a very useful property of the chromosomes at the time of meiosis. One thing is very clear: longer the or larger the region between the 2 genes, more is the probability, more is the likelihood that a crossover will occur in that region. I always give the example of the small goal of ice hockey and compare it to soccer, which has a very large hole. So, the probability of the ball getting through this goal is much more in case of soccer as compared to that of ice hockey. So, that means although it is a very crude example, but then it is effective to a child for his understanding. Hence, the crossovers are more likely to occur between genes which are much far apart on a chromosome, and less likely to occur between genes which are very close together. So, that means it is this particular frequency of new combinations which will tell us how far the genes or how close together the genes are present, and it is precisely this property which is going to help us to map the positions of the genes on the chromosomes, which will be the subject of our discussion in some of our next episodes on crossing over. And these type of maps, you would appreciate, are of paramount importance as far as the genomic analysis is concerned. Now, viewers, let us talk about Morgan's crosses of the classical linkage discovery, which ultimately also led to the corollary for the discovery of crossing over, because he himself asked certain questions on the basis of these crosses. Let us look at the screen. As far as the first of the Morgan crosses is concerned, he started his work on Drosophila and he took into consideration eye color, the character, which was purple and red. As you can see on your screens, PR plus would here mean the wild type, and the red therefore is dominant over purple, and likewise VG plus, that is normal wing length, is dominating over vestigial. So the Morgan's data was consisting of a dihybrid cross with the characters. What he found was, he performed a cross to obtain dihybrids and then followed it with a test cross. Learners, you should appreciate why he went for a test cross in the first place. Let us first look at this cross. The parental combinations, the genotypes, as you can see on your screens, is PR plus, and VG plus and VG plus as one parent and the recessive one on the other. So the F1 dihybrid which was formed was having all the flies that were having red eye color and with normal wings. So far so good. Now he took a test cross. The test cross would be between the F1 and the double recessive tester. So this is called as he took as a tester male. And then when he took this test cross, why I asked a question in the beginning, why did he took the test cross in the first place? Because we know what are the alleles of the double recessive parent. We wish to know and we wish to concentrate on what are the genetic constitutional properties of the hybrid. So that means the only recessive alleles are going to be contributed by the tester parent. So the phenotypes of the offspring will directly reveal now in the next generation in a test cross as to what are the alleles which have been actually contributed by the gametes of the dihybrid parent. So while analyzing, we can concentrate on meiosis and only on one parent, that is the dihybrid, and we can very conveniently ignore the other, and that is the tester. Please remember, this is in sharp contrast with the analysis of the progeny when we were selfing the F1 progeny in the typical conventional Mendelian crosses. So there we had two sets of meiosis to consider, one from the male parent and one from the female. Here we are considering only one parent because we are interested in the genetic constitution of that dihybrid. We already know what is the constitution of one of the males, and that is the tester males. So when Morgan performed this test cross, and what he got was a total of 2,839 individuals, he found that the parental combinations were very high in number, and the recombinants were much less. The pattern was, of course, way different from the expected ratio of 1:1:1:1:1. So that means if this ratio was not got, it clearly indicated that there was some sort of an association between certain genes. And the two of the largest classes, namely the combinations PR plus and VG plus, which would mean the red eyed and the normal wings. On the other hand, the individuals with the purple eye and the vestigial wings. These two combinations had maximum number, and these, remember, these were the combinations which were introduced from the F1 and also from the homozygous parent flies. The test cross also reveals another very interesting finding, and that finding is that when we have talked about the parental combinations, that means they were together. What about those of the non-parental combinations? The non-parental combinations were 151:154, which again meant that there was an approximately 1:1 ratio. So that means it was now very clear to Morgan that the same alleles but in a different combinations were always present. and each parent is homozygous for the dominant allele, and one gene and one of the recessive allele now is going to come together. Just to confirm this idea, Mendel performed another cross, and now he took the F1 female as the hybrid in a test cross. In this second cross, he found that when the parent and the tester male, when they were again test crossed, a different type of a result came as far as the progeny is concerned. Out of a total of 2,335 individuals, there was nothing to say that it was a 1:1:1:1 ratio. It was far deviating from the expected Mendelian ratio. However, if you look closely on the screens, you would find that here the recombinants, that is the converse of the first cross, the largest class happens to be of PR and VG plus, which is of the vestigial wings, the normal wings and the purple ones. Now, this is a recombinant, and likewise another class is of VG and PR plus, that is of the red-eyed and the normal wings. Now, this is converse to the first cross. So, that means most frequent classes again would be based on the combinations which were originally contributed to the F1 by the parental flies. So, naturally, Mendel now suggested that if this is the observation in both the cases, he was now sure that two genes in his analysis are located on the same pair of the homologous chromosomes. So, in the first analysis, we find that PR plus and VG plus are located on the same chromosome, that is, they are on the homologous chromosomes, and therefore, if we look at the pattern now, that is two pairs of alleles which are located on the same chromosomal pair, it would mean, say for example, small PR and VG, then they would be present in the F1 on the same chromosome. So, that means this we are heading towards the discovery of linkage. In the second analysis with the female parent that Morgan had made, he also presented a very common type of analysis, and that is one parental chromosome carries PR plus and VG, and the other one carries PR and VG, because we started it that way. So, there is nothing new or nothing novel in the results, because it is a very common result which is present in the general fabric of genetics. What do these results indicate? These results indicate that the genes under analysis are located on the same chromosome, and this phenomenon in common in modern parlance we like to call it as linkage or linked genes. So, that means when two genes are close together on the same chromosome, they would not like to get segregated, and therefore, they will be not assorted independently at all, and therefore, they will be called as the linked genes. And now, the point is, if everything is going to be linked, then how would the new combinations arise? Obviously, the answer is again lying in the corollary of this statement. The new combinations are going to arise from crossovers. So, that means the parental generations, the combinations, if they are remaining together because they are physically attached by the segment of the chromosome between them, it will give us a question to be asked, and that is, if all the genes were linked, then how will the new combinations arise? If nothing would change, then how would variation be coming through? So, that means how do we explain the appearance of even say minority class of non-parental combinations? Because agreed, the parental combinations are 99%, but then how do we account for 1%? So, Morgan asked this question himself and answered it too. He said that when homologous chromosomes pair in meiosis, the chromosomes may occasionally break and they may exchange parts by a process which is called as crossing over. So, crossing over also was his idea, and he said that it is the physical exchange of chromosome segments which gives new combinations, and these new combinations are called as crossover products. If you look at the screens, learners, you will find that this exchange of segments, say for example, was not there in case of linkage, because here now the two chromosomes, the two non-sister chromatids are going to exchange these segments. And now the chromosomes would change their identity genetically, because now we find that PR would go with VG plus and VG would go with PR plus. So, the new crossover chromosomes which would be formed would be now different from either of its parents. Now, with how much frequency does this take place? Where does it take place? Between which and which genes, and how frequently? These are all questions to be answered through certain numerical problems and fundamental principles of the chromosome mapping and gene mapping and recombination frequency, and also with tetrad analysis, which we would be engaging shortly. So, that means again, to define the whole process, when two genes are close together on the same chromosome, they are naturally linked and they are not going to assort independently. So, that means if they are not going to assort independently, then you would find that the recombinant frequency would always be less than 50%. So, that means if there is a recombination frequency of less than 50%, one is sure that it is a case of linkage. So, that means when there are two equally frequent recombinant classes and they are totaling less than 50%, we are talking about a linkage. And how do we define crossing over now? Crossing over can be defined as the recombination of these very linked genes which we are discussing. And how is it brought about? It is brought about as a result of interchange of the corresponding parts between the non-sister chromatids of a pair of homologous chromosomes. And how this would result? It would result in producing new combinations out of the old genes. On your screens, therefore, if we were to summarize this whole process, it would be that when we are having, if you remember on your screens, you would find that this is something like coupling and repulsion that we had talked about earlier by the hypothesis of Bateson and Punnett. So, if we are going to have these two parents, there are two situations. In the either there is crossing over. If there is no crossing over, then the genes, they would be linked and they would not be assorted independently. And if there is crossing over, then we have recombinant chromosomes. So, that means the crossing over will take place, making the linkage in a genetic makeup to be incomplete. So, that means these linked genes are not going to stay forever together on a homologous chromosome, and the non-sister chromatids can exchange frequently the fragments during the meiotic prophase. And therefore, when we find that crossing over is a characteristic feature of incompletely linked genes, if you remember, viewers, we had given you an example of incomplete linkage in Zea mays. To go through very hurriedly through this, the colored full that is in Zea mays, the colored grains as well as the full grains, they were crossed, and what we are interested here is that we got, after a test cross, we got nearly 98% of the parents and only 3.8 or 3.7% of recombinants. Now, you can talk in terms of linkage and crossing over. You can say that this is a case of incompletely linked genes. If we were to look at the non-parental combinations, there is hardly any percentage, but then it is not 0. So, that means it is not a case of complete linkage, it is a case of incomplete linkage. And 3.6% individuals which have been separated by crossing over need to be explained. So that means the products of meiosis, to summarize, would be on the basis of recombination between the genes, and we have the recombinant chromosomes 50% and non-recombinant chromosomes 50%. This would ultimately mean that if there is a crossing over, we have the parental as well as the non-parental combinations together. Don't you think it very amply justifies the statement which I had made as the opening remarks on our beginning of the series of Mendelian genetics, that is, we are like our parents yet different. So when we come to like our parents, then the genes are linked, yet different, that means some sort of crossing over has taken place. Thank you.